Welcome, Dr. Yoram
Thank you.
Richteer. Dr. Yoram Richteer, for those who don't know him, is the Chief Scientific Officer, is that right?
That is correct.
Of the Medinol company from Israel, which is currently working with Cordis and representing, which is representing Medinol products at least in Spain. So thank you very much for being with us at CSC 2018, and we will take this opportunity to talk a little bit about the products that are offered by your company, and namely the stent. Could you maybe introduce your stent, and then we'll go on feature by feature. Dr. Yoram?
Absolutely. So the ELUNIR stent, as its name, is a drug-eluting stent that is comprised of a cobalt chromium backbone, thin struts, which are particularly narrow, as we'll discuss a little bit later on, I'm sure. It is coated with a unique durable polymer that comes from a novel class of polymers, different from what has been used in the past, which elutes a rapamycin analog by the name of ridaforolimus.
Okay, well, thank you very much. I think we should go delve a little bit more in detail and look at it, each of the specific features that differentiate it. I think they do differentiate it a lot from competing products. So let's talk a little bit about the platform. I think the platform has a very interesting design and very interesting features, among others the footprint that it leaves, but not only the footprints. Could you please expand a little bit on those characteristics of the ELUNIR stent?
Absolutely, thank you. So the platform is indeed unique, both in terms of the struts themselves and in terms of the cell design. Starting with the cell design, as I'm sure most of you know, cell designs for stents have always been termed either open cell or closed cell. And those have really been the two different concepts that have dominated stent design. Well, with ELUNIR, we took this one step farther and tried very hard to marry all of the advantages that open cell designs have in terms of side branch access and conformability and deliverability with all of the advantages that closed cell designs have in terms of scaffolding and radial strength. And what we came up with is a kind of hybrid that we like to call a dynamic cell design. So the cells have a fixed size inside and outside of a curvature, but they are also very flexible at the individual cell level. In terms of the stent struts themselves, a lot of focus has been given recently to strut thickness. Thin struts, ultra-thin struts, and ELUNIR certainly belongs to that family, but in addition, we have looked not only at strut thickness, but also at strut width, because of course every strut has a thickness and a width. And what we've tried to do is to reduce the strut width as much as possible. We have what we call the narrow struts, which are 70 microns in width, and what we call the ultra narrow struts, which are 40 microns in width. And in combination, what this ends up doing is that it gives the ELUNIR stent by far the lowest metal footprint of any drug-eluting stent currently available, and we believe that this carries important advantages in terms of vascular healing following the stenting procedure.
Because of course you combine in the same structure both narrow and ultra-narrow struts, is that right?
Yes, so every ELUNIR stent is a repeating pattern of what we call alternating strut width. So you have a narrow strut, 70 microns, and an ultra-narrow strut, 40 microns, and then another narrow strut and another ultra. And so you get 70, 40, 70, 40. This in comparison to other drug-eluting stents, which typically are around 100 microns, somewhere between 90 and 100 microns in width.
Okay, thank you very much, Yoram. Further to the strut thickness and the strut with. There are other very specific features of the ELUNIR stent that differentiate it from other type of stents that we have. And from what I remember, the coating is different, and it has very specific features that maybe have some responsibility in the clinical outcomes afterwards. Could you please expand on this feature?
Sure. So, as we all know, the world started with what are known as durable polymer drug-eluting stents. And the problem is that even though they are called durable, it's only because they are not biodegradable by design, whereas in practice what happens to many of these durable polymers is that during the course of stent expansion and then drug elution, they tend to crack and peel and flake and come off of the stent, and this could have disadvantages in terms of inflammation and possibly healing in the long term. What we've done with ELUNIR is coat the stent with a novel class of polymer called an elastomer. Elastomers are polymers that have elastic properties. You can stretch them out and compress them like a rubber band. And because of this, when the stent is expanded, when drug is eluting, the polymer truly maintains its original configuration. We don't see cracks. We don't see places where the polymer peels off. Even during maximal overexpansion, the polymer integrity is maintained. And again, this cuts down on inflammation, promotes healing, and promotes a better long-term outcome for the patients.
And a better drug delivery, I suppose, a more uniform drug delivery. Okay, well...
Please.
Please. Well, let's go on with the different features. What about the drug? What can you tell me about the drug? You already mentioned it's a limus type. I mean, has it anything special that you would like to point out respecting to other limus?
So the drug is called ridaforolimus, and it's yet another rapamycin analog made by a company called Ariad Pharmaceuticals in the US. And for the most part, ridaforolimus is much like other rapamycin analogs, these so-called limus drugs, which all have a very similar mechanism of action. They all work pretty much the same. Having said that, there are fine nuances and differences between some of these drugs, and what we like to focus on is what's known as the therapeutic window, which is the ratio of how much drug you have to give in order to be effective, so to inhibit smooth muscle cell proliferation, relative to how much drug already becomes toxic and deters re-endothelialization of the blood vessel. And ridaforolimus is on the higher end of the spectrum of limus drugs in terms of having a very wide therapeutic window, again, promoting healing, enhancing safety, while at the same time ensuring maximal efficacy following the stenting procedure.
So at the end you have a more uniform, more homogeneous drug delivery with a narrower width of dispersion of concentration of drug. Is that right?
Absolutely. That is correct, and we think that all of these things come together to give you excellent clinical results.
And that is what we're coming at the end. What do we see? How do these interesting features, let me say, translate into clinical results? You have done quite a number of trials, and I know you've done some randomized trials. So maybe you could expand a little bit on these trials and explain to us what are the main results of the in vivo application of this ELUNIR stent.
Thank you. So ELUNIR has been studied extensively in clinical studies. In fact, in the latest ESC guidelines, which give recommendations to various classes of drug-eluting stents, ELUNIR was now positioned as one of the only 4 recommended drug-eluting stents in this category based on the strength of the clinical evidence. which comes first and foremost from two large studies, the Nereus study, which was conducted mostly in Europe and Israel for CE marking, together with the Bionics study, which was conducted in the US, Canada, Europe, and Israel, mostly for FDA approval, although of course the results also are extrapolable to the European Union. And what we've seen in those studies, both of which were randomized against the Resolute stent, was that the ELUNIR was found to be non-inferior. In fact, it had identical efficacy results in terms of target lesion failure. So the primary endpoint was target lesion failure at 1 year, which was an identical 5.4% for both groups. At 2 years, again, parity maintained at around 7.5%. If you look, on the other hand, at safety, which of course most of us now look at stent thrombosis, you can see at 2 years that whereas the Resolute stent had a 1% stent thrombosis rate, which by the way is a terrific result in an all-comers population, still the ELUNIR stent had 0.5, so half the stent thrombosis rate. Of course, even at 2,000 patients, those differences are not statistically significant. However, if you look at stent thrombosis confined to between 30 days all the way out to 2 years, again, there was a marked difference where the Resolute had 0.5% stent thrombosis between 30 days and 2 years, but the ELUNIR had only 0.1, so 1/5 the rate of stent thrombosis. Again, reinforcing this message that perhaps by combining all of these various elements, better control over drug elution, thinner and more importantly, more narrow struts, uniform distribution of drug, and a control over the various aspects of how a stent behaves, you can end up with perhaps a safer device.
So if there are no further remarks, I thank you very much for your explanations, for your presentation. I congratulate you for your work and for the excellent results you have obtained with the ELUNIR stent. Thank you very much, Dr. Yoram Richteer.
Thank you very much.